Here are the materials you will need to split your cells. 1 flask of cells that has previously been prepared from thawed cells. Cells will be attached to the bottom of the flask when the flask is positioned as such with the cap angled upwards. You will also have 1 empty flask. Next, you'll have a micropipetter and micropipetter tips. Here's how you insert a pipette tip into the micropipetter. In order to maintain sterile conditions, try to keep the pipette mostly covered by the wrapper until ready for use. When using the micropipetter, push the top button to pull liquid up into the pipette. Push the bottom button to dispense. When pulling up liquid, try not to let any liquid pass the pipette tip into the micropipetter. You will also be using trypsin. Trypsin is a protease that cleaves the cells from the bottom of the flask so that they are free-floating in the solution and can be transferred to another flask. Finally, you have medium. Medium, containing fetal bovine serum, antibiotics, and L-glutamine, support cell growth. The first step of your protocol is to take your empty flask and add 13 milliliters of the medium. You will later add cells to this flask, making it your final product. Next, take your flask with the cells and remove the medium and discard into the waste. In this flask, cells should be attached to the bottom, so by removing the medium, you are not actually removing many cells. Change your pipette tip. Add 1 milliliter of the trypsin to the flask. And swirl the solution, making sure the trypsin coats the entire bottom of the flask where the cells are attached. You will then remove the trypsin from the flask and discard into your waste. This step is to ensure that all the medium is removed. Change your pipette tip. Add 2 milliliters of the trypsin to the flask. And swirl the solution again, ensuring that the trypsin coats the entire bottom of the flask. Lay the flask horizontally, and then you will incubate the flask with the trypsin for 2 minutes. This is enough time to ensure that the trypsin cleaves the cells but does not damage them. After 2 minutes, remove the flask. The solution will appear cloudy once the cells have been removed from the bottom. To double check, you can look at the flask under the inverted microscope and the cells should be free-floating. Change your pipette tip. Next, you'll add the appropriate amount of fresh medium to the flask with the trypsin. For a 1 to 3 dilution, you add 4 milliliters of fresh medium to bring the final volume to 6 milliliters. This is a 1 to 3 dilution because out of the 6 total milliliters, 2 of the milliliters came from the solution with the cells. Pipette up and down to thoroughly mix the solution. You will take 2 milliliters of this mixed solution and add it to your flask containing just medium. This brings the total volume to 15 milliliters. Swirl the solution and lay the flask horizontally so that cells coat the bottom. Again, insert into the incubator and allow for cell growth. You've now completed splitting your cell.